Hi everyone, sorry for needing to miss class today. Um, uh, Normally for this lecture, which is actually one of my favorite days in the class, because we get to watch the Quiroga lecture, who was responsible for the work that led to the Jennifer Aniston neuron, which hopefully if you haven't already, when you watch the video of his about a 30-minute lecture, you'll see and hear about some of the research, but I think one of the key parts about hearing his research is hearing how he critiques it and how he explains the method and some of the potential drawbacks. As far as a researcher goes and reading his things, I really enjoy how he explains things, and also denotes the limitations. So here are the questions of the day, and given that you're probably all interested in what kind of music he would have played today, I was actually going to show you a video clip of Friends, since we're talking about the Jennifer Aniston neuron. Not so interesting, definitely not in the Iron Maiden. So let's get started, and actually before we get going on any of this information here, I think at a very, very basic level, we should all understand, especially since we've had intro and that's a prereq for this class, but we should all understand uh, how a basic neuron functions. And I think where we can start is on a picture. And you know, this is kind of the boilerplate for any sort of picture in an introductory textbook, but it's also in any sort of psych book, because there's always a biological or a biopsychological section of these. I think it's important to kind of think back to the types of neurons we have. We have sensory neurons, which are going to bring messages from the body's tissue and the sensory organs in toward the central nervous system, the brain and the spinal cord, for processing. We have motor neurons, which carry information away from the brain and spinal cord. The information coming in is afferent, A-F-F-E-R-E-N-T. Information going away from the central nervous system is efferent. And in the grand scheme of things, we've got a few million sensory neurons, a few million motor neurons, but we have billions and billions of interneurons. Interneurons are those kind of interface between other neurons, and those are definitely the most abundant and should really help you lay the foundation for understanding just how complex the internal communication network is within the nervous system. But I think that everybody should be able to look at this diagram, and honestly the best way to practice, you know, that one essay that you have, the best way to practice this is to kind of just look at the figure, walk yourself through the pathway of communication. So if we start here, because this is a good place to start, with the dendrites. Dendrites are those parts of the neuron that are going to be bringing information in. Dendrites essentially listen. They're going to be receiving information from millions and millions of neurons, bringing information in that will eventually go into the cell body of the neuron, or it's also called the soma. And as that information is coming in, whether it be excitatory information or inhibitory information, is going to be compiled or processed at what's called the axon hillock, which will be right around here. And if the excitatory messages come in, they outweigh the inhibitory messages to the point of threshold, an action potential is sent and sent down the axon. Axons can be quite long in the body and move multiple feet through the body, carrying information via the action potential all the way down toward the terminal branches of the axon. One thing to note is in places where you have myelin, which is a lipid, it kind of derived from Schwann cells, which are a special type of glial cell that wrap around the axon that essentially insulate the nerve fiber and actually speeds up communication. Because as the action potential moves down, and we'll talk about this in a second, in unmyelinated axons, it moves by the flow of ions, or charged atoms. It still does that when you have a myelin sheath, except that the information, or the charge, or the action potential, will jump from one node, which are called nodes of Ranvier, or Ranvier, and it jumps down, and there's no shifting of ions as we move across parts that are myelinated. So essentially it jumps from node to node, which is actually, the term for that is saltatory conduction. But that myelin sheath, you likely talked about this in your intro class. You have a range of speeds in these neurons, none of which are as fast as electrical current in say an extension cord, but upwards of 200 miles. Well, it ranges 200 or more miles per hour, all the way down to about 2 miles per hour in certain parts, and especially unmyelinated axons. But let's talk a little bit about what happens here as the action potential moves down. Like I said, the ions come in, and here's another one of those. You could pick up any intro textbook and see this diagram, probably just in different forms. I like this one. But if we're thinking about the direction of the action potential as we move from that axon hillock all the way down the axon toward the terminal buttons, or the terminal branches of the axon, how it's happening is when enough excitatory information or signals come in through the dendrites, and the difference between the excitatory and the inhibitory is enough to reach that threshold, an action potential is sent. Essentially what happens is the membrane of the neuron at rest actually keeps the membrane at what's called the resting membrane potential, which is essentially a asymmetrical amount of positive and negative charge on either side. So on the inside of the neuron, it's more negative than on the outside of the neuron. And so when we reach that threshold, these ion channels that are actually in the cell membrane will become open. And what that does is when they open, it permits sodium ions now to move freely across and rush into the cell, which normally they are not allowed to pass, so it's selectively permeable. And as those ion channels open and sodium starts to rush in, it's kind of like a chain reaction all the way down, and the ion channels will it's kind of like a zipper almost, if you want to think of it that way. The ion channels open as you move down, and sodium continues to rush in all the way down till you get to the end of the axon. As that's happening, you have this sodium-potassium pump where, in order for the neuron to fire again, we have to get back to resting membrane potential. So on the back end of that action potential moving down, or as it propagates down the axon, you start to see the kicking out of the positive potassium ions to repolarize the membrane as you go down. And so once you get down all the way to the end, then you get to the chemical version, where you have the release of neurotransmitters into the synapse, which is the space between neurons, and then depending on the receptor types available, that could send on an excitatory or an inhibitory signal. So again, this was just a very brief recap of that. I always think that might be a good thing to do, and I think it's important for you to be able to explain that. Again, my best recommendation would be to pick a diagram and walk yourself through the step-by-step parts, and try to be as detailed as you possibly can. So basics of neurons, I would definitely review that. Now, all of these pictures that we just talked about really, really oversimplify things, and I think it's It's interesting, though, that our nervous system is, a good way to think of it would be working in a complex fashion through a simple structure. So each one of these neurons stores and transmits information, and really it's how they eventually work together, which is some of the stuff that we will discuss later in terms of how things are working together in different pathways across the nervous system to support some of the higher cognitive functions that we sometimes take for granted in our everyday lives. But I think it would be a good point here to just really briefly touch on the basics of the neural circuits that they talk about in Chapter 2 to really set the foundation for things like excitation, inhibition, that we will discuss later, because and one of the first ones that's going to come up is something called lateral inhibition, which will be based on this idea of inhibition, but it will talk about how we will talk about how that particular process drives different perceptual capabilities. So if we think of a basic structure with these blue ovals serving as our sensory receptors, and here are our red receiving neurons, so you have the connectivity here between the sensory receptors and the receiving neurons. Here we'll focus in on the sensory receptor 2 and its connection to the receiving neuron X. And let's say that we can measure the signal that leaves that receiving neuron, which we can, of course. We would have a microelectrode. But this is probably the most simplistic structural situation that you might have in a nervous system. To be perfectly honest with you, I don't think that this is a really realistic situation. It's very unlikely that you have a one-to-one type of situation. I might imagine that you might have a one-to-one type of situation in organisms that may have a simpler nervous system. But here, a one-to-one type of situation where you stimulate here and you have a direct connection to the receiving neuron X, which sends on its signal immediately with no alteration whatsoever from any sort of neuron adjacent neurons. So the star is our light stimulus. If all of these were having light hit it, if it's a one-to-one situation, each one has its signal, and there is no modification whatsoever, given that there's no inhibition, there's no converging of neurons onto a single neuron, or no converging of multiple receptors onto a single neuron. Now, when we bring in those kinds of different situations, so for instance convergence, now if you were to stimulate just sensory receptor 2, okay, like we did the first time, you still have that direct path to the receiving neuron X. But if you think about how this is going to happen, if you stimulate 1, 2, 3, and 4, as you add on, this resulting signal is now going to be modified, because now you have the receptor hitting neuron Y, okay, which is connected to neuron X, which will further modify the signal going here. And then if we continue to add, you can see that how they all converge onto neuron X, which will all provide some input to what's going and leaving, the signal that's leaving neuron X. Now, if we think about convergence and inhibition, so now the difference here is we still have our sensory receptor 2 straight to the receiving neuron X and then the signal leaving from X. But instead of convergence happening here where the black line with the X is, you have something different going on. So now if we stimulate 2, you've got the signal straight on if nothing's happening in these other receptors. You add in 1, you still have the convergence like we did originally with 1 and 2 being active and the signal leaving X. But when you start to work in 3 and 4, now this information that's moving here to neuron Z is sending an inhibitory signal over to X. So again, it's going to modify what's coming out of X in a different way than if you had full convergence or if you had just a direct one-to-one situation from 2 to neuron X. So here's the big question. This is all very simplistic, and you can clearly see I get bored in PowerPoint. But the good question is this really a reality? Is this really something that the nervous system does, where we have these circuits where you have direct connect, you have inhibition, you have convergence? And the short answer is yes. And one of the best ways to see how that works deals with the receptive fields. And your book does a really good job of it. They start talking about some of the monkey work that you can read about in there. Some of the monkey, I think they were measuring in the forearms in some cases. But receptive fields, I mean, just like the definition implied, it's any area of a neuron's receptors that may have some sort of stimulation impinging on it impacts its firing rate. And so one of the most common types of receptive fields that we have are something called excitatory center, inhibitory surround. And given that we're all fairly accustomed to having green and red, meaning green go and red stop. So if you look at the excitatory center, the center of this receptive field is green. Any stimulation that falls in there should increase the firing rate. Then if you have stimulation that falls in the red inhibitory surround, that's going to decrease the firing rate. And so if we were to bring some light, if nothing hits within the receptive field, the receiving neuron from this, or the receiving neurons, are not going to, assuming there's a direct connect, right? They're not going to modify their firing rate because the stimulation, in this case light, is not impinging on those anywhere in the receptive field. Now, if it hits right in the middle, right, you might see a spike in the amount of activity as long as that stimulation is in the in the excitatory center. Now, if we move it, right, so if it hits the excitatory center, it's going to increase above a particular level. When we move it into the inhibitory, it's going to decrease among a particular level. So that's the excitatory center inhibitory surround structure. We also have the reverse of that in certain parts of our nervous system, where you've got an inhibitory center and an excitatory surround. Again, you have the similar situation. If the light stimulus doesn't hit within the receptive field, nothing's happening. But this time, if it hits in the center, now you've got the reverse of what's happening. Now you've got an inhibition, or a lowering of the activity, where if it hits the excitatory surround, now the green portion on the outer skirts here, now it's going to have an excitatory effect and increase the rate of a particular level of firing. So now how does this get even more complex, where you're talking, right, so what we've got here is we've got a situation where one might think it might be more akin to the one-to-one situation, but keep in mind that you've got both excitatory and inhibitory in the same receptive field, and light in this case is not going to be that, I don't know, a good word would be pretty in its hitting the parts of the receptive field. So you end up with something in many cases called center surround antagonism. And if you think about somebody being antagonistic, it's working against you, right? So if you've ever been around somebody who's working against you, they're just antagonistic. So here's an example. So if we go back, this is a situation where we've got an excitatory center, so we've got a green center, and an inhibitory surround. Now, just like we did earlier, that information hits this excitatory center. We have an excitatory signal. It enhances or increases the firing rate. Okay. If you cover up more, so the light impinges on more of the excitatory center, right, where all of it's covered, again, further increasing the firing rate. But now here becomes the situation where you have an antagonism, where you would have those circuits talking about inhibition, okay, potentially convergence depending on where it goes. So now you've got the excitatory center being covered by a light stimulus, but also some of the inhibitory surround. So now you've got excitation and inhibition all in the same situation, right? Again, working together, potentially antagonistically, and working against each other to modify the rate of firing in a particular way as a function of the stimulation. Then, of course, if the whole receptive field, both excitatory and inhibitory, is fully covered with stimulation, again, further modifying the firing rate, or the resultant firing rate. What you're going to see as we move into some of the stuff in a few lectures is situations like this allow us to see some and detect and leads to perception of very specific types of things in our environment. But the short answer to that question that I asked a while ago, does this really happen in the nervous system? Yes, it does. And as you read through the receptive field section of your book, that's a good way to try to understand and start grasping that these things happen in the nervous system. So, looking back, we should all know how neurons work. I gave a quick overview of that, but I think that you would be able to write in more detail as you move through the specific structures. We talked about excitation, inhibitions, and convergence in terms of the basic neural circuits, and we related that to receptive fields. But now here's the big question. This is the big question that I think is really neat about this area of research. It's also one of the best parts about this class as you start to explore how some of these physiological scientists are trying to make the leap from these neural circuit activities to our understanding and our putting meaning onto these stimuli that are coming in through our senses. And so this gets into the idea of sensory coding, and this was the part that I wanted to set up for our discussion, our activity and our discussion that dealt with Quiroga's work. What I wanted you all to do first, so hopefully if you're watching this before you watch the Quiroga video, I'd like you to do this. This is your ranking of accounts of sensory coding. Now your book talks about 3. I want you to try to rank which one, in terms of explanation, I guess jives with you the most. Which one do you like the most? Before you hear Quiroga explain certain things. So, do you like the specificity coding, which deals with the grandmother cells, the idea that, and you'll hear in the video him talking about Konorsky, talking about that we've got these singular cells that are kind of like identification cells. The idea of distributed coding, where it's a larger group of neurons that work together to help with the recognition process or the sensory coding of information. And then there's another form of what would be called distributed coding, but it doesn't involve such a large group of neurons. It's a sparse group of neurons. So hopefully I'm catching you before. If not, we're going to discuss this on Wednesday, your rankings of which one you like the best based on the evidence that you've read about in the book and that you've heard from Quiroga. And so we'll have our discussion at the beginning of Wednesday. Hopefully you'll follow the link that I sent in the D2L email to the 30-minute lecture. I'll say again, if you haven't already looked at it, pay close attention to what he's saying. It is a lecture, and he has a fairly thick accent for some words, so it can be tough to discern what he's saying. And the discussion of the questions, again, on the D2L email, you should have had an attachment that has questions on it that would guide you and help you walk you through the 30-minute lecture from Quiroga. I tried to keep it to the key things that I wanted to have happen in the discussion, which we'll have at the beginning of Wednesday. And hopefully we'll have some time on Wednesday. I can kind of give you a look ahead about how I think the preparation is going for the tests and what types of recommendations I would have. I will see you Wednesday.